In this podcast, and probably it will extend into the second podcast, we're going to talk about cellular chemistry, kind of the basic chemistry you need to know or to be reminded about when it comes to understanding and studying cell biology. So what we're going to talk about in this podcast, and probably the next podcast, we'll see how long it takes, is matter, what it is and what it's made of. We also want to talk about bonds. When we talk about matter, we'll talk about elements and the atoms and how those atoms come together to form bonds. We'll also talk about water, specifically when it comes to hydrogen bonds and the importance of water and the interactions it has, and how very important understanding water is to understanding cell biology. And then we're going to say just a very little bit, not much, but a little bit about acids and bases. Let's go ahead and get started with matter. Much of what we're going to cover for this section of the material is stuff that you had in Chemistry 131, you probably had it in Biology 110, and maybe some other classes, and most definitely in high school. What I want to do is just provide a brief update, a reminder of some of these important facts. First of all, the definition of matter. What is matter? In general, we can think of matter as something that takes up space, has mass. And to give a few examples here, I found this picture laying around my office, and I'm just going to put it right here. So we can look at this picture and identify the things that are matter. Things that take up space and have mass. The people have mass, so we can think of living things as having mass, and that's really what we're going to focus on in this class. So the people, of course, the grass. There's a pigeon right there that one of my kids is chasing. There's probably a whole bunch of living things in the ground that we can't see. That would be matter as well. We're going to mention a couple non-living things, because that's also matter. You see this cement column here. So the column. Looks like there's some gates there. The clothes that people are wearing. The shoes, of course. So all of this make up matter. We have living and non-living examples. For fun, if you want to take a guess and tell me where this picture was taken, I'll give you a bonus point on the next exam. As a hint, it's on a famous person's lawn. Okay, let's move back to cell biology and matter. To talk about matter, I want to talk about the smallest fundamental unit of matter. And to do that, I want to put up the periodic table. And you've seen the periodic table before. You've probably seen it more than you care to see it. But it's an important thing to go over when we're thinking about matter and we're thinking about living things. So the periodic table, I'm just going to put PT to abbreviate here. It has more than 100 different elements. These different elements combine in various manners to create matter. So all the matter we see in this picture, the people, the grass, the pigeon, it's a combination of all of these elements coming together in specific combinations to create that matter. So we're talking about these elements that will go on to make up the matter. So let's give a quick definition of elements. Elements consist of atoms with a characteristic set of protons. A lot of things could change about an atom, and it's still that element. But the number of protons cannot change. To talk about that, let's look at one of the elements here. Sorry, Jackson, I'm going to cover up your face there. No offense. And I'm going to pick one of my favorite elements. Everybody has a favorite element. So let's talk about krypton. The elements are always listed with their abbreviation. Some tables like this one spells it out. The numbers are sometimes in different places. I'm not sure why. But usually up on top is the atomic number. And this is always the number of protons. Down here, in the case of krypton, has the number 84. This is the atomic weight. And this is the number of protons plus the number of neutrons. It's the weight, essentially, of the nucleus of the atom. Protons and neutrons are in the nucleus, whereas the electrons are outside of the nucleus. So to calculate the number of neutrons then in a particular atom, you would just take the atomic weight, subtract the atomic number, so 84, subtract 36, and that equals 48. So in this atom of krypton, we would predict that there are 36 protons, 48 neutrons, and how many electrons? The number of electrons is defined as being equal to the number of protons to give it to give it a neutral charge. So we would say there are 36 electrons. The one thing that can't change in this number are the number of protons. If you change the number of protons and you make it 35 protons, it then becomes bromine. So it becomes a new element. The number of neutrons, that will change to some extent. And if you held a handful of krypton atoms, not every one of those atoms are going to have 48 neutrons. There will be a mixture. There will be some with 48, some with fewer, some with less. But that's the average number. And these different forms of krypton with the different numbers of neutrons, those are called isotopes. You can also change the number of electrons. And if you change the electrons, you will change the ultimate charge of the atom. And when you do this, it's still krypton, just like when you change krypton's neutrons, it's still krypton, it's just a different isotope of it. When you change the number of electrons in an element like krypton, it becomes an ion. Okay, I'm going to erase this, and I want to spend a little bit more time talking about the electrons of the element, of the atom, to start talking about bonds. And I'm going to talk about carbon now, because it's an easier element to draw, to be honest with you. It's also a very important element when we start talking about cell biology and life in general. So if we look at carbon here, it's right here, it has 6 here and 12 here. So that means carbon has 6 protons and 6 electrons. We really want to focus on the electrons now. In the nucleus of this atom, as I said before, you will have 6 protons and you will have 6 neutrons. I didn't explain it when I had the numbers just written here, but remember that bottom number was 12, so 12 subtract 6 gives you 6 neutrons. It has 6 protons, so it has 6 electrons. And though I'm not going to really focus too much on the different energy levels of these of the valence shells here, I do want to point out a few things. We have 2 electrons here, and then the next one will have 4 for a total of 6. And I'm just going to draw them like so. These electrons in the outer shell, so electrons in outer shell, help determine the atom's reactivity. It is these electrons in the outer shell that will interact with other elements to form bonds. When 2 atoms come together, they will form a molecule. Molecules, a simple definition would just be two or more atoms that are chemically joined. Some examples would include oxygen, carbon dioxide, a little larger molecule like ATP, or amino acids. These are all molecules. Now there is a special category of molecules called compounds. This is a molecule, as I said before, it's a special kind of molecule with two or more different atoms. So up here, the molecules that would also be compounds would be CO2, because you have carbon and oxygen. ATP, I didn't draw out the structure, but there's more than one kind of atom in ATP. Same thing as with amino acids. The only one up here that is not a compound is oxygen, because it contains only oxygen. One way you could say this is that all compounds are molecules, but not all molecules are compounds. This is a molecule, but it's not a compound. But all these molecules, and many others that we didn't write here, of course, are compounds. Now that we're thinking about molecules and compounds, let's talk about bonds. What's holding these atoms together to form the molecules and bonds? So we'll talk about bonds. And the bonds we're going to talk about are going to be covalent, ionic, hydrogen, and van der Waals. And in case I forget to mention, I wrote these in the degree of strength of the bonds. So covalent bonds are stronger than ionic bonds, which are stronger than hydrogen bonds, which are stronger than van der Waals bonds. And what I mean by stronger, I mean the individual bonds are stronger. One could argue that collectively, all the hydrogen bonds together are very strong. But when I say bond strength, I'm talking about a single bond, a single hydrogen bond versus a single covalent bond. So let's talk about covalent bonds. So covalent bonds are strong bonds that share, and this is the key word, share electrons between two atoms. A good example of this, and to be honest with you, it's a good example because it's easy to draw, is if we take hydrogen. Each hydrogen atom has one electron. The covalent bond would have these two electrons being shared. So they're sharing each other's electrons to form this covalent bond. An ionic bond, let's talk about that next. Let's separate these here so we don't get them confused. To define an ionic bond, it's a bond, it's also a strong bond, but not as strong as a covalent bond, that forms due to the transfer of electrons. So for instance, let's talk about our favorite ionic bond, sodium chloride. How does sodium chloride form? Well, sodium here has 11 electrons, and chloride has 17 electrons. The sodium will lose an electron, and it will form this sodium now only has 10 electrons. Now it's going to be less negatively charged because we just lost an electron. Then the same number of protons are there, so this is going to now be more positively charged. Chloride atoms, which have 17 electrons, they will gain that electron. So now the chloride atom will be negatively charged. It will have 18 electrons. This electron over here is lost and given to the chloride. So now we have a negatively charged chloride. the negatively charged chloride and the positively charged sodium will now combine, because they just transferred an electron, to form sodium chloride. So that was all ionic bonds, and I ran out of room to talk about the others, but that's just as well, because I want to skip hydrogen right now and just spend just a little bit of time talking about van der Waals. I want to give you a brief definition of van der Waals, but I'm not going to spend a whole lot of time on it. These interactions, van der Waals interactions, due to weak electrical attractions between two nonpolar molecules. I think of these changes in these electrical attractions as transient changes of these charges. Singly, they're very weak, but together, van der Waals attractions lead to very strong interactions. For instance, it's the classic example is thinking of the gecko that can climb straight up a glass window. It's because of these van der Waals interactions. Okay, I'm going to clear some space here so I can spend a little bit more time talking about hydrogen bonds. We can define these as weak interactions, weak bonds. So singly, they're weak bonds, but remember, together they're very strong, between two molecules from an electrostatic attraction. Hydrogen bonds don't always occur between water, but we often think about water when we think about hydrogen bonds. But remember, it's not just water that involves hydrogen bonds. Though in my example, I am going to talk about water. So if I were to draw a very simple water molecule here, it can bind with a neighboring water molecule, and this water molecule can bind to an adjacent water molecule. And I'll draw one or two more here. And this can go on and on and on. In fact, each of these hydrogens will bind to their own water molecule, same with that oxygen. And the reason they can do this is that they take advantage of partial positive and negative charges. The hydrogens have a partial positive charge, the oxygens have a partial negative charge. All of them do. And so that allows for this very weak bond between these oxygen and hydrogen molecules. There's a couple terms I'd like to reintroduce you to as we move forward, and they are the terms of cohesion and adhesion. Cohesion essentially are when like molecules interact. So, for our purposes, what we're talking about often when we say cohesion is when two water molecules interact. Adhesion means when different molecules interact. For our purposes, we can think of like a water molecule interacting with a non-water molecule. A good example of this is when we think of a tree. So this is my tree. It's lovely. And to get the water molecules to move up this tree, the water molecules have to interact with each other. They form a chain. So that's cohesion, water molecules interacting with each other. This also provides an example of adhesion. The water molecules stick together for cohesion, but how do they move up here? They are required to hold on to these vascular structures that then move them up there. So we have water molecules also binding with these vascular structures. We'll come back to water molecules every now and then, and hydrogen bonds. The next thing I want to remind you of and bring back to your attention are some terms that we associate with solutions. If we had say a beaker here with water, we would call this an aqueous for water, and solvent. Solvent is the term that we use to describe the substance that material is being dissolved into. So we're going to add to this aqueous solvent a solute. That is the substance that will be dissolved into our solvent, in this case an aqueous solvent. And then what we have, once it is dissolved, we have the solution. Now seems like as good as time as any to talk about the terms hydrophobic and hydrophilic. These are very important interactions in cell biology, and we can define the hydrophobic interactions as when non-polar, NP for short, molecules, or it could be regions of a larger molecule. You will have a large protein, but there are some regions of it that will be nonpolar and some regions that will be polar. So when these nonpolar molecules or regions will aggregate with each other when in an aqueous solution. In essence, what this does is it keeps the nonpolar regions together with water surrounding it. Often we're told hydrophobic means, because that's kind of the Latin root words, as being water-fearing. You could say that, but it doesn't really tell you what it's doing. This is a better way to describe it because it's telling you what the molecules are doing. And I think as a good example of that is to think about a membrane. And we'll spend a lot of time talking about membranes. You'll love membranes because we're going to spend that much time talking about them. But they're formed between these phospholipids. And I'm not going to draw this all the way out, but each one of these polar heads here, these are polar, and then these are nonpolar hydrophobic tails. These interactions are really important. They aggregate together, as we say here, they aggregate together, and they exclude the water. The water is all out here. And it interacts with these polar parts, providing this sort of boundary around, an aqueous boundary around the membrane, keeping these nonpolar tails aggregated and away from the water. I don't know if they really fear it. I think they're just kind of forced to do it. They might very well like water. I don't want to pass judgment on them. So what's hydrophilic? This is simply just usually polar molecules interacting with water. So hydrophilic would be this interaction right here. Water interacting with these polar parts of the phospholipids. There's just one more thing I want to cover, and that includes bases and acids. The best way to introduce this at this kind of introductory level here is to talk about our pH scale. I'm going to make this one down here 1, and this is 14. So pH is anywhere between 1 and 14. Near the top represents bases. So something with a high pH is going to be a base. As a definition, bases acquire protons Or you can think of them as giving up electrons during a reaction. Counter that are the acids, and they're going to be our lower numbers. Acids, they give up protons, or you can think of it as accepting electrons during a reaction. So anything, let's make our neutral. 7 is a neutral pH. Anything below 7, we're going to call it an acid. Anything above 7, we're going to call it base. And 7, we're just going to call it neutral. If you had something at pH 6 and you wanted to make it higher, if you wanted to go from a pH 6 to a pH 8, you would add base. Now sometimes, to make sure that the pH doesn't change much, we could add a buffer. And buffers will kind of block some of the pH changes. And this is very important in cell biology research and a lot of scientific research, so that we don't have large changes of pHs. Alright, that's all I had to say, so I'd like to close with a summary. Over this unit of material, we talked about several things. We talked about matter. We mainly talked about living matter, but we talked about how matter is made up of atoms. We talked about the subatomic parts of an atom, the protons, the neutrons, and of course the electrons. We talked about how to interpret or how to calculate how many protons, neutrons, or electrons there are in an atom. We defined isotopes. We talked about electrons and how they establish reactivity. We then talked about bonds and how these electrons, introduced here, are important in forming covalent bonds. And remember, with covalent bonds, we said the key word there is that they share an electron. And then the ionic bonds were the next strongest, and they transfer an electron. That's a key difference between these two. And then we talked about hydrogen bonds and how hydrogen bonds form due to the partial charges of the water molecule or of the hydrogen atoms. And we talked about these great things called van der Waals interactions. And we talked about how these are very transient charges, but they're very strong. They allow things like the gecko lizard climbing up a very smooth glass window. And we talked about water and solutions and the role that water has in life. We talked about a couple examples with hydrophobic and hydrophilic, and we talked about adhesion and cohesion. So water is very important, and we're going to come back to that often. And then we closed with a very brief discussion of acids and bases and pHs. That's all I have for this podcast. So if you have any questions at all, please make sure you stop by and I'll be happy to help you. I'll see you in class. Bye.